how can we evaluate spatial resolution for EEG and MEG in a way that's intuitive and useful for experimenters, that helps us with the interpretation of our data and is not too theoretical and abstract. Well, I will deal with linear methods here. Linear methods are probably the most common ones for source estimation, minimum norm type methods and beamformers, also called spatial filtering methods sometimes. And we talked about the basics in a different session. I just want to remind everybody the basics, the basic concept for what we are going to talk about are the point spread function and crosstalk functions. Point spread functions for a given source at a certain vertex or voxel in the brain tell us how activity from a source here with unit strength would spread to other brain areas. So this would be a simulated source here. So you compute the forward solution, you then subtract it to your linear source estimation method of choice. You see what it does with it. So how does it spread out this activity? You will never get just this focal source back here. And then what you see here, that's the point spread. And the whole distribution is called the point spread function. And in related concept, very useful as well, is the crosstalk function. So imagine you're only interested in activity from this particular source. You don't really care about other sources, but that's exactly why you want to make sure that other sources don't leak too much into the estimate for this location here. So you want to know how much do other sources leak into this estimate. And this is given by the crosstalk function. So how much crosstalk is there from potentially uninteresting, non-relevant sources to the source of interest? Now you can, of course, go around the brain and plot these for all your dipoles in the brain, or at least for the regions of interests that are relevant to you. But if you want to do that for at least a significant subsample of the whole cortex, then you will spend a lot of time doing it. And also it's even harder than to compare different methods if you want to do it source by source. So it's usually much better to find some numbers that summarize these functions like localization accuracy and spatial extent, which you can then plot across the whole cortical surface and potentially compare across methods. And of course, this has been done. But before I show some of those examples, I mean, just to highlight what I mean. So the two most common measures that I think have been used in the literature to compare methods or evaluate spatial resolution are the localization error and spatial extent. So imagine you have a point spread or crosstalk function like this here in just one dimension. It's usually not doing us the favor to be nicely Gaussian. So, the just average or standard deviation usually don't, they don't have the same interpretation, of course, like with the Gaussian function. So it's usually a bit more annoying. So you have a peak somewhere which is somewhere different from the true source position. So that would be localization error. Here we go. And it also has a spatial extent. It has side lobes. It goes up and down, positive, negative. But even so, you also want to find a measure for the spatial dispersion. But just keep in mind that, it might not be exactly like a standard deviation for a Gaussian function, but the formula is basically spatial deviation in space. So that's what we call spatial dispersion here. And it's important to note that in order to evaluate spatial resolution, it's not just peak localization that counts. So it's interesting to know whether the peak is in the right location or how far away it is from the right location. And often people talk about dipole localization only even in methods comparisons. So they seem to think the only metric that is relevant is this peak localization. However, the spatial extent of these point spread and crosstalk functions matters as well, because as soon as you have more than one source, the spatial extent will determine whether they will blur into each other or whether you can separate them from each other. So this is just briefly illustrated here. So imagine you have your two dipole sources close by in different locations. If you only have one dipole source at a time, that's fine. So you just take the peak and it accurately tracks your dipole. But as soon as you have them active simultaneously, they just blur into each other and they are not distinguishable from each other. So it's both dipole localization error as well as spatial deviation or the spatial extent that matter. And this has been used or was used, for example, in this study to compare EEG and MEG, or actually to demonstrate the benefit of combining EEG and MEG together. So here we see peak localization error computed for every source across the cortex. So dipole by dipole, you compute the point spread function, which in the case of MNE The minimum norm method is actually the same as for the has the same shape as the crosstalk function, so it applies to both. So you do that for every source, and you can plot the distribution for EEG alone. Trying to remember how many electrodes they had, I think about 70. Might not be exactly true. Then for MEG, there will be 306 sensors, and then for the combination of EEG and MEG. And they also had a condition where they actually matched the number of MEG and EEG sensors to have a fair comparison that doesn't depend on the number of sensors, which is usually different between EEG and MEG. And in this case, high dipole localization error obviously is undesirable. So white and yellow colors here indicate a high localization error, and darker colors a low localization error. So darker colors are better. And in this case, you can clearly see MEG compared to EEG wins, so lower localization error. But if you combine EEG and MEG, it actually looks very similar to the MEG alone. So in this case, adding EEG to MEG actually doesn't help, at least not very much. Localization error stays the same. It's mostly determined by MEG. So you might conclude from that it's not necessary to get your hands dirty and glue these EEG electrodes before the MEG experiment. However, if you look at the spatial extent, the spatial deviation measure, you see a different pattern. So again, larger numbers are bad because that means a more widespread point spread or crosstalk function. And if you look at EEG alone, well, you get some relatively quite high numbers here, especially in areas that are probably not well covered by the EEG electrodes. MEG looks different, looks a bit better. So it has lower numbers across a larger part of the cortex. It also can't really get these numbers down in depth. And if you combine EEG and MEG, then you see that this looks even better, I would say, than MEG alone or EEG alone. So in this case, adding EEG to MEG results in improved spatial resolution. So that is the clear result from this study. So in summary, localization error doesn't change much if you add EEG to MEG, but spatial extent, spatial resolution actually improves, which means as soon as if you're dealing with more complex sources rather than just focal dipoles, then I think combining EEG and MEG is definitely advantageous. Okay, this is actually just a reminder. We had this on our, we talked about this in another session. So this just reflects what we already knew from our sensor space analysis. If you look at sensitivity maps, so if you compare MEG and EEG, yes, I mean, they are sensitive to different sources. And if you combine them, you get more information than when you look at EEG or MEG alone. And this is reflected in the spatial resolution as measured by the spatial deviation in the previous slide. So there's clearly a correspondence here, I think, between what we actually see in our sensor space data and what we can then do with our source space analysis. Okay, so we, shameless self-advertising here. So we used measures like that and an approach like that to compare different source estimation methods. So we looked at combined EEG and MEG measurements in this case, so to basically look at the best case scenario. So this is the vector view, the MEG and vector view system plus 70 EEG electrodes applied to the Wakeman and Hansen open data set. So this was done in a group of, basically as a group study. So we computed these measures for several subjects, and we are presenting the average. And we compared a number of linear methods, the most common ones. I mean, the classic L2 minimum norm estimation, a depth-weighted L2 minimum norm estimation, dynamic statistical parametric mapping, DSPM, exact and standardized Loretta. And we also used two LCMV beamformers with different covariance matrices. More about that later. And at this point, you don't really need to understand the theory behind these methods. So no matter where these methods come from and what their mathematics is, we have our metrics, localization error and spatial deviation. So we want to know whether the point spread and crosstalk functions look good to us. And if that's the case, then we know the method does what we want it to do. And if not, then we might actually find something better. So the resolution metrics in this case, as I said now several times, peak localization error and spatial deviation. Yes, but before we actually look at these values, we check out some individual point spread and crosstalk functions. I think that's always a good idea. If you know which regions of interest you're interested in, then it's always nice to visualize these to get an idea of what spatial resolution you've got. So in this case, well, it looks a bit complicated, but hopefully it will make sense. In a second, we have 3 regions of interest, maybe hard to see, but there is a little boundary here that is the anterior temporal lobe. Then there is the pars triangularis here, and the pars opercularis. So they are in all of these panels, highlighted in all of these panels here. And we computed point spread and crosstalk functions for these regions. And for MNE, as I mentioned before a few times, so these point spread and crosstalk functions are the same. So if you plot one, you got the other one for free. So you can interpret these distributions both ways. And in this case, we are comparing that with E Loretta. But for E Loretta, point spread and crosstalk are different. So we have to plot them separately, as you can see here. And we do that now for these 3 different regions of interest separately. So this would be for the anterior temporal lobe, this column here, and we can now compare our methods. And in this case, if we look at MNE, well, generally speaking, we can say whatever happens in anterior temporal lobe stays in anterior temporal lobe. In the point spread interpretation, if there is a source in anterior temporal lobe, the activity is more or less confined to the region of interest that we outlined here. However, there is some leakage to outside this region as well, but at least it's close by. I don't see very much here in the different lobe, here in the inferior frontal lobe. If we now look at the point spread function for E Loretta, we can see that it's actually a bit more widespread. So the peaks are also in the anterior temporal lobe in the region that we chose in the region of interest. But it is a bit more widespread now, and it also looks like there's some blue here down in the Sylvian fissure. And I should also highlight that E Loretta has 0 dipole localization error. So that means the peak of this distribution is exactly in the right spot where the original source is, at least if we look at point sources. However, we can already see here that compared to MNE, we have a bit more spatial extent. And if we then look at the crosstalk function, then the eagle-eyed among us might realize that this is actually very similar to the crosstalk function here also for MNE, and that's not by chance, because the way these methods are designed, with the Loretta methods basically being a normalized version of the classic L2 MNE estimator, the crosstalk functions are actually exactly the same for these 2 methods and also other linear methods that I'm going to present. So that's a property these methods have. So you might choose different methods like DSPM or E or S Loretta, and you will get a different point spread function, but actually the crosstalk functions will stay the same. I may talk about this in a different session at some point. I'll need to think about that. Okay, let's look at another region of interest now, pars triangularis here, actually very similar story. With MNE, you mostly have your activity within or around the pars triangularis, little leakage to the neighboring region here. For E Loretta, it looks similar, but just as before, a bit more widespread, maybe a bit more leakage into the other neighboring region. The same again for the crosstalk function, we have the same as for MNE. But finally, for pars opercularis here, it looks a bit worse. In this case, activity is more widespread. It actually spreads also into the neighboring region, so it would contaminate here activity in the pars triangularis as well. If we look at, and this is particularly relevant if you wanted to compute connectivity, for example, between these 2 regions. So if this region leaks into this region and you find significant connectivity, it might just be that it's activity that only comes from pars opercularis, but because it leaks into this region and the neighboring region, you see some significant connectivity. We will have to talk about this kind of these type of confounds when we talk about functional connectivity later. E Loretta doesn't solve this problem. So if anything, it's a bit worse. Again, slightly more spatially extended point spread function here. And the crosstalk function looks the same, or at least very similar in this case to the PSF and CTF here. I'm afraid the scaling is, I think, a little bit different. So that's why you can't really, it doesn't look exactly the same here. But now we can compute, just like we did with the comparison between, how the other group did it with the comparison between EEG, MEG, and the combination of EEG and MEG. We can compute our peak localization error source by source across the cortex and plot them as distributions and compare them across each other, across different methods. So this is like quite a complicated slide. So I can only refer the interested listener to the paper where this is described in a bit more detail, but hopefully I can get the main messages across here. So we have peak localization error plotted here for point spread functions and for crosstalk functions. So we compute these distributions vertex by vertex, and then we compute the peak localization error and we plot it. And we do that for the different methods like MNE, depth-weighted MNE, DSPM, S, and E Loretta. Not surprisingly, for MNE, for the point spread and crosstalk function, it's exactly the same because the two functions have exactly the same shape. And all these methods have been suggested in the literature to deal with the kind of localization bias that you get with MNE. So MNE would normally localize sources that are deep to areas that are more closer to the senses. I mean, that's just what the minimum norm method does. So there's some systematic mislocalization here, and other methods like depth weighting have been suggested to alleviate this bias a little bit and to improve localization error in deeper brain areas. This indeed works for depth weighting for the point spread function, but interestingly, the crosstalk function doesn't really budge. It's again similar or even the same to the classical MNE method. DSPM is a noise normalized version of MNE, also supposed to improve localization accuracy, which it does for point spread functions. So you have a lower localization error for point spread functions, but the crosstalk functions are exactly the same as for the classical MNE. And now S and E Loretta both have been designed to produce zero dipole localization error. And they do exactly show this property here. So localization error for point spread functions is zero everywhere, even in deep brain regions. But as soon as you look at the crosstalk functions, it is, again, exactly the same as for the classical MNE and all the other methods. And that is already clear from the theory that we haven't really talked about yet. And now if we look at the spatial deviation, a measure for the spatial extent of these point spread and crosstalk functions, well, long story short, none of them even gets close to zero. So these spatial deviation, I mean, these point spread and crosstalk functions will always have a spatial extent. They will never be, just a little single dipole somewhere. So it goes up to even higher than 5 centimeters here, especially if you go into deeper areas. And it's also quite clear the lack of information that we get as further we go away from the senses. None of these methods can compensate for that. So you will always get much higher spatial extent if you go deeper into the Sylvian-Fisher, for example, or in the south side. We also see that here for the peak localization error, by the way. None of these methods gives you small localization errors for the crosstalk functions in deeper areas. So there will always be systematic leakage from deeper areas, actually more from superficial areas into deeper areas, if you define regions of interest in depth. But for the spatial deviation, it's quite clear we can never go even close to zero. And it certainly gets worse if we go to deeper brain areas. And well, some methods are slightly worse than others. For the CTFs, they all look the same, again, because the shape of these CTFs is the same for these methods. For point spread functions, you see some differences here, but I would even say just by eyeballing, the darkest red is actually here for the classic minimum norm estimator. So all the other methods do improve localization error to some degree, even down to zero localization error for the point spread functions, but they do not change the crosstalk functions. And also for the spatial deviation for the point spread functions, if anything, they even make things worse. So long story short, you may gain some in terms of localization error, but you may lose some in terms of spatial resolution here, especially when you look at the spatial deviation measure and the crosstalk functions. Okay, we can do something very similar for beamformers now, which are a bit of a different kind of beast. But yeah, I haven't talked about the theory yet. So here we can just look at what, in practice, what do they do in terms of point spread function and crosstalk functions. It's a complicated slide, so I'm using different regions of interest here to illustrate the differences between different types of beamformers and MNE. So here I'm using the lateral occipital cortex as one region and an area possibly reflecting around the auditory cortex and posterior superior temporal cortex. You see quite a few images here because we have point spread functions and crosstalk functions, but also the beamformers actually depend on the data. So not just on the noise from baseline measures, but they actually use the data covariance matrix to localize sources. So you're basically projecting, you're going around, it's a bit like a dipole scan, where at every location you check how much the data, the forward solution of a dipole projects on the measured data. It's a bit more complicated than that, but that's more or less, I think, what they are doing. So we can compute the data covariance in different ways, and correct ways, and in not so correct ways. So in this case, I actually compared beamformers when I compute the beamformer based on evoked data from a visual condition, so the visual checkerboards that we use in the MNE data sample data set, and beamformers with the covariance matrix for the auditory condition. Long story short, actually, that doesn't make a big difference. So we can actually ignore the different auditory and visual condition for the sake of this presentation. Also, what we see is that point spread and crosstalk functions don't seem to differ that much here across these conditions. I also have a beamformer computed with a covariance matrix from the baseline interval that also doesn't change very much. So that's the good news, actually. The different beamformers from different data sets don't seem to differ that much. However, we can see that these are quite widespread. So we have quite spatially extended point spread as well as crosstalk functions. And if we compare that to the MNE down here, then we can see that MNE actually seems to have a more focal distribution, certainly in visual cortex, but I think also for the auditory region of interest here. And well, if we would look at the theory in a bit more detail, I don't find it very surprising. So you might remember from a previous session that MNE is actually the method that optimizes the spatial resolution matrix. So if you tell it minimize the spatial extent of point spread and crosstalk functions, that's what you will get. Well, this kind of constraint is not part of the beamforming method. So the beamformers have a different optimization criterion, which you may or may not find useful for yourself, but you just have to accept that beamformers do not optimize the resolution matrix and point spread and crosstalk. And you can see that here. So crosstalk and point spread functions are more widely distributed. And this also shows up when we look at the resolution metrics again and plot them across the brain surface. And again, I computed in this case, two beamformers here, one for the baseline interval, so pre-stimulus interval, one for the actually proper data covariant, based on the proper data covariance matrix for the post-stimulus interval. They don't differ very much. But for this beamformer here, you actually have zero dipole localization error for the point spread function, a bit like the Loretta methods, which is great. But as soon as you look at the crosstalk functions, you can see that you have quite a large spatial extent for these large spatial extent for these crosstalk functions. Also, for the other beamformer, you have slightly higher localization error here. So this is really just, well, you can see a shade of red here because it's just, the covariance matrix are very similar. This is, but also for the crosstalk function here, you can see quite a large spatial extent in most brain areas, especially if you go deeper into the brain. And also here the scale is different, like from the minimum norm type estimators. So there I cut it off at 5 centimeters. Here I had to cut it off at 8 centimeters to make, so that we can actually see some differences here. So it's clearly larger than for the MNE type methods. If we look at the spatial deviation, again, for our pre-stimulus beamformer, post-stimulus beamformer, the differences are not very big. Between these types of images, between these images here, they actually look almost identical, I would say. And again, the spatial deviation is really huge. So the spatial extent of these point spread and crosstalk functions is very high across the whole cortex, and I think it's clearly larger than for the minimum norm type methods. Okay, so I've shown you what you can do with these metrics, and also just with point spread and crosstalk functions, how to visualize them, how to get numbers out of them. I mean, if you don't agree with the way I've done it, then you can do this yourself. So these methods are available in MNE Python. But for me, the conclusions from this methods comparison are that methods vary quite a lot, actually, with respect to localization error and spatial deviation. So that means that there is a choice to be made for us. However, I mean, improvements in localization error are accompanied by increases in spatial deviation. So there is no method that really optimizes everything. And if there was a method like that, then we would have solved the inverse problem. And we knew that the inverse problem is underdetermined and non-unique and all the rest of it. So there simply cannot be any clever mathematics that gives us information that is not in the data. We can only make different types of compromises. So if you improve one thing, then something else might get worse. You know, it's a bit like a balloon. You poke one side and something on the other side will stick out, which is actually not what happens on the balloon, but anyway, I like it. Localization error for point spread functions can be minimized. So there are methods that have zero localization error for point spread functions. E&S Loretta, and also some types of beamformers, depending on the covariance matrix. But this is not achievable for crosstalk functions. And there are very good reasons for that, which I haven't really explained based on the structure of the lead fields. So yeah, don't be fooled if people tell you that, oh, their method has zero localization error, because there are still the crosstalk functions and they won't budge. You cannot, I mean, if you look at the spatial extent of both point spread and crosstalk functions, you simply cannot get below a certain point. So there will always be some spatial extent. So always some leakage between sources. You can only choose basically where it's worst and maybe where it's best. So you can choose different methods that maybe move this leakage, this kind of distribution around a little bit, but you will never find a method that really optimizes everything perfectly. And we looked at beamformer separately from MNE type methods. So what I just said also applies to beamformers. You can get localization error for point spread functions that is even zero or close to zero, but the spatial deviation in general for both point spread and crosstalk is clearly higher than for MNE type methods. So I would say that therefore that they have lower spatial resolution than MNE type methods. I also looked at beamformers, or we looked at beamformers with different covariance matrices, and they seem to be pretty similar. I mean, this was all computed on for one data set. So obviously you may, I mean, this might differ if you look at resting state activation or patients versus controls. I mean, that's where you might have to pay a bit more attention. But at least for this kind of experiment, event-related experiment, I'm actually quite reassured that results obtained for different types of beamformers in the past are relatively generalizable to other situations. But there is still potentially a problem with beamformers that, you know, one beamformer, my beamformer might be different from your beamformer, depending on what kind of data I'm using and how I'm computing my covariance matrix. Well, an easy conclusion is there is no obvious best method. I'm afraid, you know, I can't really recommend anything specific here. So in this analysis, a bit more details in the paper, I would say the classic MNE and E-Loretta seem to offer the best compromise between localization and spatial deviation. I find MNE easier to interpret. E-Loretta is a bit more complicated. So the simplest is usually the best. So in my personal preference, therefore, would be the classic MNE method. But the tools that I've used here for this comparison are available. So they are on MNE Python. You can compute resolution metrics and the point spread and crosstalk functions. Look at them yourself. So I can only strongly recommend you to actually try this yourself. Thank you very much.